right, so DNA replication in prokaryotes. The good thing about this is that it is very similar in eukaryotes. There's a couple differences, and we'll talk about those tomorrow. But if you get this down pat, you'll be pretty smooth sailing for tomorrow. So, prokaryote DNA has been well studied because it has a relatively small genome, and there's a large variety of mutants, making it easy to study. So, like we said the other day, E. coli is the most studied prokaryote DNA. There's 4.6 million base pairs in one single circular chromosome. That single chromosome can replicate its DNA in about 42 minutes, so less than an hour. It starts at one single site, so you know it's circular DNA. So then it starts at one site, and then it will proceed in both directions instead of just going one direction until it gets back to that starting point. Approximately 1,000 nucleotides are added per second. So this is very, very quick, and there's very, very few mistakes made in this whole process. So the biggest player in DNA replication is DNA polymerase. This is the enzyme that adds nucleotides one by one to the growing nucleotide chain. Does the exact same thing as RNA polymerase did in protein synthesis. So this addition of nucleotides requires energy, and this energy is obtained from ATP, GTP, TTP, or CTP. These are all your nucleotides: ATP, adenosine, adenine triphosphate, GTP, guanine triphosphate. And so as those add to the growing nucleotide chain, it loses one of those phosphate groups because it gives up that energy. That energy then is used to attach the nucleotide, which would be ADP then, to the growing DNA strand.
Wait, so it loses energy after so many get added, one drops and then it loses?
Yep. So remember, ATP has 3 phosphate groups bonded together. So all of these, that's what the T is for triphosphate. And so let's say there's an adenine on this one. If this adenine nucleotide needs to get added to the DNA, that will break off this phosphate, which releases energy. And then this energy that got released is used to attach this adenine diphosphate to the DNA molecule. Okay. You might see the abbreviation NTPs. NTPs are nucleoside triphosphates, which are simply these 4 guys up here. It is the generic term that doesn't distinguish whether it's adenine, guanine, thymine, or cytosine that's attached. It's just saying that it's that one of those nucleotides with 3 phosphates. They're high energy molecules, and they serve as the source of the DNA nucleotide and the energy. So like I said, the energy releases when that breaks off, but then it's instantly used to attach the nucleotide to the growing DNA strand. When the bond between the phosphate is broken, so that would be this step right here, breaking that bond, energy is released and then used to form the phosphodiester bond between the incoming nucleotide and the growing chain. So remember, the bond that holds the nucleotide to the DNA, that's that phosphodiester bond. So the energy released helps make that phosphodiester bond. All right, so a little complication when we talked about RNA polymerase, it was just RNA polymerase. There was only one, whereas DNA polymerase has 3 varieties, which we call DNA polymerase 1, 2, and 3. POL, abbreviation for polymerase. So each of these DNA polymerases has a different job. So DNA polymerase 3 is required for DNA synthesis. DNA polymerase 1 is important in DNA replication. and DNA polymerase II is required to repair your DNA. So there'll be diagrams up in the future slides here with these sorts of labels on them, DNA Pol 1, 2, or 3, indicating where they are in the process of making DNA. All right, so the beginning of this replication, it starts at the origins of replication. My brain says that term makes sense. Origin means beginning. So this is where replication begins. And this site is recognized by certain proteins. Those proteins will bind to that site, which initiates the replication process. So again, E. coli has one origin of replication. There are other bacteria that have multiple origins of replication. When your human DNA replicates, it starts at multiple locations. It doesn't just start at one. This origin of replication is about 245 base pairs long in E. coli, and there is a lot of AT sequences, so adenine thymine sequences. If you were to look at it all sequenced out and you saw AT, that would indicate to you that that maybe is where that origin of replication is located. Okay. So once the origin of replication is recognized by those proteins, another protein, this is a protein, helicase. Ooh, it's probably an enzyme, which is a protein anyways, so that's fine. Helicase's job is to untwist the DNA or unwind it. This will break the hydrogen bonds between the base pairs, which requires ATP hydrolysis. Remember, hydrolysis deals with water? Mm-hmm. And so you get a water molecule at the end.
Doesn't it break the water molecule? Or no? Never mind.
Yes, it would break the water molecule. Hydro for water, lyse to break or cut. Good. So once the helicase has done its job, what you are going to see is a replication fork, which is what I have the diagram in here for. The replication fork, there are two of them. So here's one, and here would be the second one. This is a structure that forms as DNA begins to open up. There are two formed at the origin of replication. That allows your DNA synthesis to extend bidirectionally. And that's kind of indicated here by your arrows, one moving towards the top and the second one moving down. Two directionally, two directions. And then we get these single strand binding proteins that show up during replication, and these are proteins that bind to a single strand of DNA, and this helps keep the two from joining back together. They're kind of like little separators that just come in to hold the two strands apart. There's a different diagram, maybe on the next slide, 2 slides away, that will show those single strand binding proteins. So again, DNA polymerase is really great and good, but it's got 2 restrictions. The first restriction is that it can only add nucleotides in the 5' to 3' direction. And so this we'll take a look at on the next slide when we have the diagram, so I can explain that a little better. And then the second restriction with DNA polymerase is that it requires a free 3' hydroxyl group. I think we talked about that maybe last week on Friday, about how you need the free hydroxyl group there in order to add the nucleotides. When we talked about the DDNTs, the ones that block it, because they don't have the free hydroxyl group there. So having that free hydroxyl group allows for the formation of the phosphodiester bonds between the 3' hydroxyl and the 5' phosphate of the next nucleotide. You cannot add nucleotides if there is no free 3' hydroxyl group. So this becomes a problem because that very first nucleotide that you need to add is not going to have a free 3' hydroxyl group. So you need the assistance of a primer. And the primer provides that free 3' hydroxyl group. So what happens is RNA primase is going to come in, and it's going to make a strand of RNA that's going to attach to the DNA, about 5 to 10 nucleotides long. This is going to serve as a complementary template, or to the template DNA. So then the primer is that RNA sequence right here that RNA primase made that primes the DNA synthesis. So it's a little bit before the section of DNA that's getting replicated. And now DNA polymerase extends beyond this RNA primer. It adds the nucleotides one by one, and it's complementary to the template strand. So all of this we can see here on this diagram. So first thing we want to look at that we talked about is helicase. Helicase's job is to unwind the DNA. Then we talked about these little purple guys here. Those are that single strand binding protein that help hold the two strands of DNA apart. So at the top strand, what we're going to focus on right now are the RNA primer and the primase. So the RNA primer is building strands of RNA. So the RNA is, remember, would have a uracil instead of a thymine in there. And it's building it because this primase, this blue ring, is telling it to. Because you can't add a DNA nucleotide because there's no free 3' hydroxyl group on, let's say, this nucleotide. So that primase and the RNA primer need to provide that hydroxyl group. Once that hydroxyl group has been provided, your DNA polymerase 3 will come through and read, all right, this nucleotide is an adenine, so I need a thymine. So it will attach a thymine right there. Okay, so again, if this was a guanine on the template strand, then DNA polymerase 3 would say, I need a cytosine. And the cytosine would come from the cytoplasm of the cell. Does that make sense? Okay. And so that DNA polymerase is going to go through reading all of those bases from the template strand. The first restriction of DNA polymerase said that it has to add from the 5' end to the 3' end. So this 3' end is from the original DNA strand. And this 5' end is also from the original DNA strand. So when the DNA polymerase comes through, let's look at this bottom one. Since this parent strand is 3', that means that the new strand has to be starting on the 5' end. Does that make sense? Because they run anti-parallel. So that means that on this bottom strand, that DNA polymerase 3 is going to move towards the replication fork because it has to add from the 5' to the 3' end. Does that make sense? So then on the top strand, since the original was a 5', that means the bottom strand on this far edge is going to be 3'. So that means the DNA polymerase is going to add in this direction, away from the replication fork. The next slide then. All right, so if you noticed on that diagram, there was labels of the leading and the lagging strands. So a couple things before we get to that, I guess. The replication fork will move at a rate of 1,000 nucleotides per second. So in this diagram on this slide, this replication fork is moving to the left because of that label right there. Okay, so it's going to open up as you move to the left. There is another enzyme, topoisomerase, that's going to prevent the overwinding of DNA helix ahead of the replication fork.
Isn't that the green?
Yep, it was the green. Good. So this kind of just holds it together. So you know when you twist two ropes together, if it's tied at one end, this end's going to get really super twisted and it just doesn't work. And so this topoisomerase pretty much just clamps it. It's a movable clamp, but so that it can't twist. This prevents twisting further up. That topoisomerase is going to cause temporary nicks in that DNA helix, but those nicks, they're kind of like little gaps, actually, might be a more better visual term. They will get resealed later on. So there's a problem with this DNA replication. And the problem is that the two template DNA strands, they have that opposite orientation. One goes 3' to 5', the other is going to go 5' to 3'. And so that creates something called the leading and the lagging strand. So the leading one is up here down, and the lagging is on the bottom. So the leading strand is the strand of complementary DNA, and it's complementary to the 3' to 5' strand. So here's the 3' to 5' parent strand, and it's complementary because it adds bases on the 5' to 3', which is the way it's supposed to add, right? 5' to 3'. So the leading strand can just zip along continuously. It doesn't run into any hiccups because it's smooth from that 5' to 3'. That's the natural way. The lagging strand is the strand that is replicated in short fragments. So those fragments are in red down here. They are synthesized away from the replication fork. And what happens is because of the requirement to add from 5' to 3', it needs a little assistance. And so it needs this RNA primer every time it starts a new segment. So that RNA primer gets laid down, and then the DNA polymerase can come in and create the new DNA strand. And it will happen in short little fragments. And those are called the Okazaki fragments because for once there's a Japanese scientist studying this stuff. I feel like we're really good with the British and the American scientists. Or European scientists, but now we get a Japanese guy doing this work. And so what he found is that on that lagging strand, the DNA gets added in short chunks. And that does slow it down a little bit. And so that's why it's called the lagging strand, because it's lagging behind that leading strand. And so, let's see. You'll notice then that there are now little gaps between the two new DNA segments. Yeah, that would be a nick or a gap, yes. And those will need to get closed up. So, some finishing touches. The sliding clamp is a protein that holds the DNA polymerase in place as it continues to add nucleotides, making sure it doesn't slide off the DNA molecule. It's a ring-shaped protein. It binds to the DNA, and it holds that polymerase in place. It's kind of a connecting port, so to speak. As DNA synthesis proceeds, the RNA primers are replaced by DNA. That would be on the lagging strand. So back here where we've got those green RNA primers, eventually they will be replaced by DNA. Here's kind of the process. So the first step is those primers are going to get removed by an exonuclease activity of DNA polymerase 1. So DNA polymerase 1 is going to come in and pretty much cut out those primers. exo meaning out. Then it will use DNA from behind that RNA as its own primer. So this second one over here is maybe a better example. So let's use purple. So once this green primer is going to get removed, then DNA polymerase 1 will come back to the previous Okazaki fragment and use that Okazaki fragment as the primer to fill in this gap. Okay. So that was step 2. This fills the gaps left by removing the RNA nucleotides by adding DNA nucleotides. So DNA ligase is another enzyme that helps build the phosphodiester bonds between the 3' hydroxyl end of one nucleotide and the 5' phosphate end of the other fragment. It's used to close the gaps between the newly synthesized DNA segments. So we get the 2 segments that are made, but then this DNA ligase is going to come in and glue them together. It acts as that binding agent because it helps create this phosphodiester bond. Does that make sense? So this step puts the missing nucleotides down, and this DNA ligase then comes in to glue or connect the 2 DNA fragments, that finishing last bond that needs to be there. So again, this DNA polymerase 1, here it's coming through to close up the gaps. And then your DNA ligase is then gluing the 2 fragments together. Otherwise, I think your sliding clamp is up here. Otherwise, I think...
It'd be really nice if they'd go in order and not all over the place.
What do you mean by go in order?
Like, the helicase is the first, right?
Yep.
And then it's the DNA polymerase 3.
Yep. So they put it on both strands instead of just one to kind of spread it out so it's easier to read. But I understand what you're saying, but now it doesn't really flow in order.
Yeah, that's...
So that lagging strand has a lot more work that needs to be done to it versus the leading strand. The leading strand pretty much gets that DNA polymerase, that sliding clamp in place, and it just goes. It can do its thing. It doesn't need to worry about the 5', 3' thing because it's just set up to be successful that way. Whereas that lagging strand, that really has a lot more things going on.
So it's like a rich kid to a poor kid.
Pretty much.
Oh my God. I'm not trying to be mean.
So here is a summary of everything in about 10 steps of what happens in this DNA replication. It's a good summary, but remember that's just it. It is a summary. Some of the details are missing and would make this is a great first step for studying, but you will need the details come test time. So step 1, that DNA unwinds at the origin of replication. The helicase opens the DNA and forms those replication forks, and they extend bidirectionally. The single-strand binding proteins coat DNA around the replication fork to prevent the rewinding of DNA. Topoisomerase bonds at the region ahead of the replication fork to prevent supercoiling. Then we have primase that will synthesize RNA primers complementary to the DNA strand. Again, is that on the lagging strand or the leading strand?
Wait.
Step 5.
Lagging.
The lagging, very good. Then DNA polymerase 3 starts adding nucleotides to the 3' hydroxyl end of the primer. Then the elongation of both the lagging and the leading strands continue. The RNA primers are removed by the exonuclease activity. The gaps are filled by DNA polymerase 1 by adding dNTPs, those DNA nucleotides. And then the gaps between the 2 DNA fragments are sealed by DNA ligase, which helps in the formation of those phosphodiester bonds. So again, very simplified, but it does give you at least a picture of that. This table, you know how I like my tables. This gives you the enzymes and the proteins involved, and then what they do. So this makes for a nice matchy-matchy on a test or a quiz. It also helps you keep enzymes organized in your little brains.